Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. We are from Group 5. We will be presenting our material about biopesticide manufacturing process. Our group members are Rizal, Zaka, Nurhakim with study number 143, Clarisa Sheiladiva with study number 178, Yehezkiel Aldi A.M with study number 198, and Rumampuk Yunita A with study number 199. The first is definition of biopesticide. Biopesticide is a pesticide that is made and taken from natural materials. According to Junaidi, 2009, biological pesticides are formulations containing certain microbes in the form of fungi, bacteria, or viruses that are antagonistic to other microbes or produce certain compounds that are toxic to pests. Biological pesticides are included in environmentally friendly pesticides because they have an impact on plants in a certain period of time as well as easy to degrade. Residue is quickly lost, doesn't pollute environment, and safe for living things. Study 2022.
Function of biological pesticide. According to Indiati, biological pesticides have various functions as a repellent, rejecting the presence of insects because of the strong smell sting. And the next is antivedant, which prevent insects from eating plants that has been sprayed because there is a bitter taste. And then nerve poison and attractant, attracting the presence of insects in the trap insect. In addition, biological pesticide also functions to prevent insects from laying eggs. This disrupt the hormonal system in the insect body. The last one is control the growth of fungi and bacteria.
Okay, so the next is advantages and disadvantages of biological pesticides. Advantages of biological pesticides. Biopesticides have the following advantages, such as first, quickly decomposes due to sunlight, air, humidity, or other natural components. This characteristic causes the risk of contamination reduce land and water. Residual risks on crop yields too low. Second is toxicity in mammals is generally low, so it is safe for humans and livestock. Third is high selectivity because biological pesticides have a spectrum extensive control. And the last is phytotoxicity. Phytotoxicity is low, so that the potential for plant-based pesticides is damaging low plant. The next is disadvantages of biological pesticides. According to Sino 2016, weaknesses in biological pesticides such as first, slow working ability so that the result of spraying biological pesticides are not visible in a short time. Second, doesn't kill pests directly, but will reduce appetite for pests so that pests will die slowly or just to repel or make the pests don't want to eating plants that have been sprayed with biological pesticides. Third, easily damaged and easily decompose when exposed to sunlight. Fourth is can store for a long time, so its necessary use after completion and becomes an obstacle to trade to the public. And the last is it needs regular application, so it is not very efficient in terms of economy.
Material and tools. Materials used in this activity is the pure isolates of biological agent various types. Potato dextrose agar or PTA or seborrheic dextrose agar or CDA. Aqueous, spirits, alcohol, and LCL, blotting paper, roll tissue, aluminum foil, gauze, gas LPG, plastic, cotton, and others. Other material can be replaced with corn rice. The next is tools. The equipment used in an autoclave isolation box, Bunsen lamp, microscope, object glass, slide glass, petri dish, Erlenmeyer, test tubes, dissecting sets, multi-purpose pen, plastic basin, scales, small spray, filter, marker, hector, plastic trays, and storage shelves.
The next is the procedure of manufacturing biopesticide. There are two procedures in the process of making biological pesticides. The first one is making biological agent starter. The process of making biological agent starter can be started from steam corn or rice with steamer for 30 minutes. The second one is spreading corn on the tray until cool. Next, enter each 100 grams into a plastic bag, and the result of packaging can be re-entered to a bigger plastic. And the next is sterilize the material in the autoclave at a temperature of 121 Celsius degrees with a pressure of 15 C and kept constant for 60 minutes. Next, after sterilization, the material is removed and cooled. The next one is pure isolates of biological agents were inoculated using an ose needle in a corn rice media. Next, the plastic is removed and shaken so that the fungal spores can be spread evenly on the media. Next, incubate at room temperature for more and less 2 weeks. And the starter ready to be distributed and used for propagation at the farm level. Okay, the next procedure is the how to compounding biopesticide. Step 1 is mushroom inoculum is added with water and kneaded until the spores are released, then filtered and into the sprayer. Step 2, the dose is 4 until 8 kilograms of mixed inoculum with 250 until 400 liters of water for a land and an area of 1 hectare. Step 3, applications should be made on morning or evening by a sprayer. And the last step for Bassiana mushroom spraying should be directed on the plant crown. As for Trichoderma, it should be sprinkled around the plant roots.
Okay, thank you for your attention. If you guys have any question, you can ask us on Spider.